So how will synthetic biology and new materials change the way that we manufacture key products such as pharmaceuticals, food, electronics, consumer goods, and more? That's the question, and in order to answer that question, we have, and I'm going to introduce the new panelists first. Meredith, maybe you want to say a few words because you haven't been on stage yet, but she's a partner with partners. You know, you are the first, I guess, pure play investor here on stage today. Maybe you want to say a couple of words, and then I'll move on to the other new panelists, and then we will take it from there.
Sure. Do you want? I was going to say by way of background.
Yeah, just
okay.
Introduce yourself,
sure.
and then I'll give you a question.
I'm a microbiologist by training, so this is always an exciting opportunity to talk about this. But actually, an investor by practice. Partner at Partners Innovation Fund. We are the venture capital arm of Partners Healthcare. So even though I'm at MIT, which is actually also an alma mater for me, so I'm happy to be here, I actually represent investment interests in putting money to work in new companies on the basis of inventions coming out of the partner system. And in fact, the speaker that we just had before us is actually operating on some technology out of one of the PIs that we have. So terrific to see them doing so well and really exciting. Partners focuses on putting money to work, as I said, in new technologies. And so we look for opportunities where there are materials, medicines, as well as devices, and even diagnostics and software that can be part of the life science ecosystem, both within the pharmaceutical industry and beyond.
So before I move to Malik, it just strikes me that since some people have just come in, this is Vincent Ling of Takeda, was just speaking. We had Christine Santos from Manus Bio, one of the MIT startups that was on stage earlier. We have Professor Collins, of course, of the Collins Lab, also involved in a major amount of the startups presented here today. And then lastly, Malik Tucker from Novartis. And why don't you say a few words? So we have called you a lab head of synthetic biology group, but there's a lot to that. So just give us a little sense of what you and Novartis are up to in this little field, and then we will get started.
Sure. So I'm Malik Thakur. I'm a molecular biologist by training. I'm a lab head in synthetic biology group at Novartis. So Novartis is, I believe, one of the first major pharma to start a dedicated synthetic biology group. And this was started around 4 years ago. And we are still a small presence here, but we have other colleagues in Basel, which is our another site in Switzerland, where around 30 colleagues are involved in working on natural products, and we are trying to merge this synthetic biology and natural products to expedite the research that we can do in natural products discovery. They are still early days for us. We are looking for partners. We are really open to there are search and evaluation teams that are constantly looking and evaluating various startups, and we have some collaborations already going on with MIT. We had a project with Tim Lu's lab earlier. We have an ongoing collaboration with Chris White. It's with Foundry, which is MIT, Harvard, UCSF, and Broad put together, which is headed by Chris White. That's a multi-million-dollar collaboration that we have for 5 years. So things are looking really awesome right now.
Perfect. Yeah. So I'm gonna throw the first question at Professor Collins, because you obviously spoke earlier and you were sort of talking about your work. But if you were to say, what should we, as an audience of a grand we, be the most excited about, and how should we structure this conversation about the application field that's opening up in synthetic biology? And remember, this discussion is being recorded so I can show it to my 10-year-old daughter. This is sort of gone are the slides and all of that stuff. We need to speak, hopefully, here at a level that everyone can understand. So, you know, if you were explaining this, you know, maybe to a 10-year-old, what are the applications and what should they be focused on going forward?
Yeah, so I guess maybe I'll start with a cautionary comment, and that is that I think we have to bear in mind that it's very early in this field, and it's really hard to engineer biology. So we hear the success stories this morning, but most of the stuff that my lab tries to do doesn't work. And it takes us a long time to get what we want to work. And so when you face that coupled with the hype that surrounds this field, it's often difficult to discriminate between what's reality, what's the potential, and what's just science fiction. And I think this field more than any has been troubled by that. Having said that, I think it's an incredibly exciting time. The field is not only young, it's small. So I think we also get media coverage that well outpaces the actual size. And to point to this, iGEM, which is an undergrad competition and program in synthetic biology, has more teams than there are active academic labs in the world. And so it speaks to that we're lagging behind. Having said that, if I were talking to a 10-year-old, and I talk to a lot of 10-year-olds, is that I think that synthetic biology will be a defining technology of this century. It may not be the defining technology of this decade. I think machine learning will be that. But I think it will be a defining technology of this century. And I think that we can turn to the power and diversity of biology to address, in part, many of the world's big problems. And that includes health, food and water, energy, and the environment. I think the challenges are huge, but I think bringing the engineering perspective to biology, which is really at the heart of what synthetic biology is about, is exciting. So on the applications, obviously, in the health is a space I focus on. I think it's very early in this space, but the ability to harness other organisms and biological components to both sense is there something wrong in the patient's body and to also remediate, I think is tremendously exciting. So this notion of having living medicines, living therapeutics and living diagnostics is very early this decade, but I think will become a dominant theme the next decade and the decade after that.
So I've had the blessing of having this feeling, and I don't know if this happens to you, but I get goosebumps when I really know that I'm in the right place at the right time, and I think I'm part of something exciting. We have had several of these moments, and I'm wondering if there's more of you that, the statement you made is pretty striking. You think it's going to be basically the foundational technology of the next decade. Let's move to Christine and hear. So you are, obviously on the commercializing side of this. You've been in the lab, and now you're really on the lab floor and trying to commercialize this. What is the feeling inside of a startup right now about the excitement, the potential? Does it feel like that when you are doing it? Does it feel like everyone is starting to realize what Professor Collins just said, or does it feel earlier than that to you from a startup's perspective?
I mean, I think definitely, this on?
Should be.
So, and definitely for us, we see a lot of potential for this technology. We're really in the realm of chemical manufacturing, and the potential is enormous if you think about all of the different products that you use every day now that contains ingredients that can be accessed through this technology. So, for example, who here brushed their teeth this morning and used toothpaste with the mint flavor, or who here uses laundry detergents that have incorporated a lot of these fragrances that are sourced from nature. That's what this technology enables. It's essentially the middle class population has been growing. It's expected to more than double within the next decade, and there's been an enormous pressure on being able to find sourcing for the ingredients that are used in a lot of these products. And our goal is really to be able to leverage these technologies, leverage fermentation-based processes to be able to access these natural ingredients and make them scalable, accessible, and affordable for not just a subset of the population, but really for the entire world, especially as we see this growth in the middle class.
But Christine's Manus Bio has been at this for a few years. Was the understanding, and we're going to move to partners in a while, but was and is the understanding in the investment community at the level that Jim is presenting here? Did a lot of people, when you came out with Manus Bio, a few years back, was this understanding that this is now ripe for massive sort of commercialization? Would you say that has sort of evolved over the last few years, or has I guess the investment community, from your perspective, seen this already, and have they started adjusting for the fact that this perhaps is the new platform technology of the century?
I mean, absolutely. Even before Manus Bio was founded, there are companies kind of exploring this space. But I think we're now just starting to see the fruits of that labor, with the commercialization, for example, of bio-based polymers from lactate acid fermentations and 1,3-PDO. And so, I think it's just the beginning. Certainly, it's a very challenging problem, but it's absolutely ripe for innovation.
Mm-hmm. Vincent, you have made some of your thoughts known to the public here a few minutes ago on the market situation and how you have to exploit niches to be able to act in this space as a startup, but perhaps also actually as a large pharma. Because the challenge, and that struck me, in what you said, I mean, the difference really isn't that great from my perspective. If you sort of think about all that we have heard today, it seems to me, and we'll have Novartis, address this later, but it seems to me that a company any size would be daunted by hearing that we're dealing with a technology that is the platform technology of a century. Because who is a winner in that 10 years, 15, 20, 25 years from now? Can you address that challenge from the perspective of a large company that obviously wants to grow, but has to reckon with the fact that Synlogic here has grown up in 2 years and is listed on NASDAQ. And who knows how many other companies will be listed on NASDAQ before many large companies even say A, B, or C.
That's right, yeah. So the speed of discovery certainly is faster in a startup, less bureaucracy. I enjoy the startup world, been in many startups myself, I've made some myself. In a big company though, it has the reach to develop drugs, and that's something that you can't do very well in a small startup. So, for example, right, when people start talking about new biological systems and we go to JP Morgan, partnering conferences, a lot of biotechs come to us and start talking about their platform technology. And on one hand, you can take the business approach and say, well, this has a lot of potential. Okay, let's see what we can do with this and see where it takes us, because it's a large addressable market. On the other hand, there's a certain pragmatic approach too. We know that it takes upwards of 15 years from inception to getting a drug on the market, right? And that's daunting. Another thing that we have to keep in mind is that in pharmaceutical, in big pharma, usually the chief executives change over once every 5 years, and once that happens, all projects are at risk because the new guy wants to put a stamp and make it as individualized, portfolio of projects going through. And that dislodges a lot of things along the way. So these major shakeups happen once every 5 years. And so in the back of my mind, when I talk about this, I think, is this feasible in a time frame that we can execute on, all right? There's a time notion there too. And timing makes all the difference in the world. Right now, really hot are CAR T cells and things like that, and microbiome is very, very hot. I agree, it's a lot of promise. But how long will it take for this to get to market? And how large the clinical trial does it have to be? Does it use FDA-approved products? Because if you introduce too many new things in the process, then you're inventing not just one drug, but an entire process that has to go through the FDA.
But isn't that part of the point, and this maybe brings us into a can of worms, but I mean, isn't that part of the point here that in this synthetic biofield, not everything is going to be FDA approved, right? So some of there's nutraceutical spaces. There's a lot of opportunity because of the area itself. I mean, this brings us, I think, to something I want to get to later, but I just want to let me get to the end of the row. Meredith, can you address some of the, I don't know, the fears and also the certainty in the investment community and in your community about what this is bringing to us, and what goes through your mind when you're going through these opportunities? What is it that entices you about it? Because clearly, it's not all certain.
Sure.
And some experiments do fail.
A lot of it, yes, most. I think Jim hit the nail on the head, right? The way I think about it is actually maybe a little bit different. I think we can look at the past, maybe in the past 10 years, because for instance, about 10 years ago, when there was an era of biofuels and biomaterial companies that were somewhat all the rage from the investment community. And what they ran into was part of what Jim said, which is biology is hard. And then I think part of what Vincent said, which is the time horizon for products is long. And then I would fold in a couple other things on that, which is price, volume, and industry, because those three things actually all have to align in order to be successful within a marketplace. And so I think what Christina was alluding to, which is sort of a flavor and fragrance arena, is one where you have a higher price per volume. You have a different speed of being able to get into the marketplace. You have a longer time horizon for interest in a product, unlike a nutraceutical, where those are very trend-driven. and you have a different type of regulatory body than you do in the FDA, right? And so I think those are the types of things in the way that we actually need to think about it, because if we're back to what Novartis is doing, which is looking at developing perhaps new forms of natural compounds, things like that, that's terrific. But I'll throw it to you two. How much is a single new molecule in your screening library worth to you at this moment? And does it make sense for me as an early stage startup to make a whole bunch of those molecules, because I need to understand how much you guys are going to pay me for those, how long it's going to take for those potentially to get to market, how challenging it might be within sort of a new, let's call it, regulatory regime that has to react to things like that, that actually really are important factors in making decisions about how to actually put money to work and when to put money to work into startups.
I don't know if you very quickly want to address that.
Yeah, so let me step back and try to summarize what we've been talking in the panel and even all the presentations. And so if you try to look at a picture as a bullseye, then at the most periphery are the flavors, fragrances, and food industry. And then if you get one level inside, then you get nutraceuticals and probiotics. And then if you come even nearer to the bullseye, then you have, say, the diagnostics. And at the core is the pharmaceuticals. And I think this is what we are trying to summarize, right? And it is all from creating the niche to the most needy requirements. The core is when we say pharma, then it's the need. We need these medicines. The periphery is not something we need. We are creating niche. The fragrance or flavor, there are other alternatives, whereas for the core, there are no other alternatives. The other things that we probably touched upon was going outside to inside. What we see is the stakes are higher, the risk is high, and it takes a longer time to develop a molecule in that bullseye going out from out to inside. And the approval is also starts getting stricter and stricter. So if you are a startup, just getting back to your question, if you are a startup, then probably this is the high risk area if you are at the center or the core of the business. But if you start from the periphery and then start getting in, then probably that's the best way to get in. That's how I'll
So we're going to soon bring in the audience, because I promised. But I have one little quick thing that I want you to all handle. So we were talking here about as if there is sort of a center and periphery discussion. But if you think about some of the maybe hidden things and maybe somewhat away from the drug use case, but what are some of the ways that synthetic biology is already affecting our lives? And if each of you just can come up with an example or something that I may not think about, or the audience is maybe not thinking about, that's already becoming either an experimental part of something that I should care about, or actually is already embedded. Okay. And maybe we start this way. Meredith, do you have an example?
I would note the non-GMO movement that I think is quite interesting in the food and probably in flavor and fragrance, because one of the things that's sort of up for debate at this moment is whether something that is actually manufactured by an engineered organism, separated from that organism, and then delivered into a product qualifies as being GMO, genetically modified organism. And I think that that's an interesting one. If any of you are at Whole Foods, you'll see that stamp everywhere. And something that'll be quite interesting for us, because I think synthetic biology is going to be pushing more and more and more food products into the marketplace, and sort of how that is actually handled with the pushback perhaps on things being considered non-natural is an interesting one.
That's a great point, and it brings us to something. We'll see what the audience questions takes us into, but it certainly brings up the dimension of Europe and other places where
Yes,
that is a
absolutely.
massive,
Huge,
even
more
so we haven't
challenging.
even
Yeah.
broached that topic yet, but great. Vincent, a quick point?
Yeah, sure. My PhD is actually in plant biology. It's not in pharmaceutical science, right? And so from a plant biology background, it's interesting that, like you said, how does engineered things affect our daily lives? I'd like to point out that most, if not all, the natural foods that we eat are engineered already by plant breeding, okay? Wheat is probably one of the most engineered crop plants you can think of. It's allohexaploid, comes from 6 different parents, 8,000 years of history there. You know, the genetics behind that is phenomenal, right? And you just don't do it by using genetic engineering. You just do it by breeding. But it's synthetic. The regular wheat that we eat is not real. Okay, and so a lot of people don't understand this. So there's an educational process too. Like, is GMO really bad?
Yeah.
Huh. Christine?
I mean, I think what's interesting is that, although synthetic biology is seen as a relative new field, and there's resistance in some areas to, concepts like GMO, like these processes have been around for decades. Fermentation, production of enzymes and products they're already embedded in things that you use in your daily lives. Biotech enzymes such as proteases and lipases are incorporated into your laundry detergents. You have, again, as I mentioned before, bio-based polymers from lactate acid fermentations and 1,3-propanediol that are in your apparel, carpets, diapers, food packaging, plastic utensils. I mean, it's basically ubiquitous already. And so I do believe that there needs to be kind of an education process to get consumers to understand that this is something that has been around for a long time and has made enormous contributions to daily life.
Perfect. We may pick up on all these, but my promise is looking very thin here. So let's quickly get on to...
So I'll go quickly. I don't think the field is embedded yet. I think it's too young. But I think we are beginning to see tools and intermediate capabilities being introduced, both in research as well as impacting industries where you may never see a product. So Ginkgo, has multiple partnerships where they're engineering organisms for companies that are using those to make things. So you'll never see the Ginkgo product, but it's buried. But there are some examples. So I'm involved in one, Sample 6, that actually has a food diagnostic that's approved and in 70 different facilities around the world doing rapid food pathogen detection and already impacting a number of industries to keep you healthy.
Perfect.
Yeah, I'll give you a very specific example of a recent drug launched by Novartis called CTL019, marketed by a different name, Chimera. So that's a first CAR-T cell therapy which has been approved by FDA. And what they do there is, this is for pediatric ALL, basically leukemia, cancer, blood cancer in kids. And what they do is they take the patient's T cell and they bring it to the lab. They use viruses to re-engineer them and put it back into the patients so that now these T cells can recognize the cancer cells and they eat them away. So this is kind of synthetic biology, right? They are being used.
I love the beauty of the example too, because
yeah.
actually I think a 10-year-old can grasp that concept. This is wonderful. Let's see what the audience has to say. Please shoot. Any question is good, and any question is legit. Anyone have something to kick it off? All right.
Shall I stand up? Maybe for people.
Yes, and lift that up. That's the side of the mic. Yeah.
Perfect. So up even
Yeah.
more.
It's a microphone.
Good?
It just looks like a box.
For the 10-year-olds, it's a great toy. So a great discussion so far, excellent. Maybe a point that people may be thinking about, and we a little bit with the GMO relationship thought about it as well, the ethics, reaction to this area. But then, some of the comments made me think, what is really natural and what is really synthetic? If bacteria in the nature changing something, it's okay. But if a human being is thinking about it and changing it with some tools, is it not okay? So I'm curious how people are really thinking about it.
Thanks, Erdogan. This was Erdogan from GE Ventures. Anyone want to pick that up? What is natural and what is not? I think, Meredith, you were touching on it, but does anyone want to pick that up? It's an important discussion, I think, that we can't hide from.
Yeah, I mean, I guess the question comes down to who actually gets to define that and who should be defining that, because I think, again, within each industry, there are probably going to be needs for a regulatory body to set that standard, as well as also sort of an industry body, and then I think importantly a consumer body as well. And some of that probably comes back to what we were talking about earlier, which is the level of education and understanding that each of those groups of let's call them stakeholders need in order to make that definition qualified for the way that I think we're evolving in our understanding here. And it'll be interesting to see how that continues to play out moving forward.
Jim?
Let me make one comment that I think it's an important distinction, but probably not the right one. And that is, I think the right one is safe. Are you introducing something that's safe? And why I say that is that I work in antibiotics, and I've had the FBI in my office multiple times concerned about synthetic biology, and are we and others going to make something that's dangerous? And I keep telling them, it's not us you need to worry about, it's nature. And that nature makes nasty stuff. And we're going to lose hundreds of thousands of people from natural stuff. And so it's really safe versus unsafe. And in our Synlogic example, where they're moving their trials underway, FDA's embraced what they've done, in part because they're actually introducing natural components in a synthetic way into the environment, endowing it with new capabilities. So it really gets after safety, obviously also efficacy. There's a question.
Yep. All right, let's see.
You can do it.
You ready?
Wow.
Keep watching.
They might need you in New Orleans on Sunday. That was good.
So at Ginkgo, this is a really important topic about how the industry can accept GMO or GMO-derived ingredients. Our CEO has actually written a New York Times op-ed on this, and we're very pro-transparency. I think in the past, sometimes industry would sweep certain things under their rug because for whatever worry. So we're very pro-transparency in whether something is made with GMO or not. And the other thing is when you're moving, therapies are a little bit different. Therapies, GMO, you're going to take, you're sick, you're going to do it. But when you're looking more at consumer products, it's really, it doesn't matter. We can in this room talk about how it's safe and it makes sense and everyone probably agrees, but it's really the consumer public that we have to convince, and so there, oftentimes it's almost an emotional response, and so that's something that we think about. We have a team basically dedicated to that because I do think it's very important in the industry as it expands to different, in synthetic biology as it expands to different industries.
Who wants to pick that up? It was a little bit more of a comment, but is there someone who?
I would like to add something to it. So when we say GMO, it's a very negative term, and but there's a positive side to this conversation as well, and which is about chemical versus natural. And there's an example, I'm not sure whether it's Ginkgo or somebody else, they are into vanillin. Vanillin, they are trying to engineer yeast or bacteria to produce vanillin. Now, if you look at that chemical, it's like a phenolic aldehyde. It's the simplest chemical to synthesize, and that's the reason we see it being added commercially available. You have vanilla flavor for everything because it's a very cheap molecule to make. Then why are we going this way, using biology to synthesize them? It's because anything that has that chemically derived vanillin goes to Walmart, whereas if you make it through biology, you can label it as all natural and it can go to Whole Foods.
Yes,
right?
that's right.
So,
Yeah.
so that's a big positive on that end.
Just to punctuate two comments that have been made that I think are very important. And I go back to therapeutics. So, the impact of synthetic biology and therapeutics, I think, is going to be at two levels. One, natural products and diversity have been hampered by deconvolution and synthesis. And then now we have biosynthesis and predictability. And that has hampered a lot the use of natural products. That was mentioned by the lady and by others, and I think that's going to be fundamental. And second, cell therapies have been with us, but they haven't been with us really for the last, 15 years? They didn't take off. And they didn't take off because of potency and because of measuring pharmacology. And these are the two things, and manufacturing. And these are the three things that potency, pharmacology, and manufacturing that synthetic biology can bring to that, carefully, of course. But these are two things that I'm not sure were said, but as the young synthetic biologists that we want to be, these are three things or four things for therapeutics that are going to be critically enabled, I believe.
Thank you.
Yeah, so maybe I should add, in a pharmaceutical company, people look at the impact of what your therapy brings, okay? So even if it's very exotic, if the payoff is huge, right, they'll invest in it, right? So for example, CAR T, I think is fairly, academically, it's interesting and it works, but I think scaling it up so it's affordable to everyone is going to be it's going to be a huge task, right? But the payoff is huge as well too, and so people will go into that, as you can see.
I really enjoyed it. I wanted to ask a question, though. It seems like the sky's the limit in terms of the natural drugs that we can produce nowadays. But if we think about oral administration, yes, we'll get to the colon, could be colonized or not, maybe needed or not. Some metabolites will get to the liver, the kidney. Are there other diseases where you think the delivery will be a limiting aspect in which you want to get your drug, your natural drug, to a specific area? Will then the delivery be important because you can use then potentially materials as a depot, as a way to use adhesive, for example, for localized delivery as a depot for release of stuff over time? So you may not need to take them on a daily basis. So some of this aspect of, okay, we can overcome manufacturing, we have a range of drugs we can now deliver, we can now produce. How do we deliver them and get them to the right place in the right time?
All right, so anyone? So Vincent, who else wants to address this? Yep.
Okay, so what you're getting at when I was showing you Lupron Depot, right? It came out first as a 1 month depot, 3 month. Now there's 6 months, right? The 6 month depot took 10 years to develop. Okay, the other ones, of course, led up to that, right? But as you see, the payoff was huge. So having depot drugs is a very large addressable market.
I'd say blood-brain barrier is one that's interesting. There's a lot of interesting new work that's coming out, even by, of course, some researchers within our system, looking at natural metabolites that are created in the gut by our microbiome that actually are conduit across the, let's call it the gut-brain axis, that actually are crossing the blood-brain barrier and appear to have implications for neural health in many different types of either disease states or actually aging states. That's quite an interesting one. And then I would actually say the other area that I think is also quite interesting and or sort of other areas that are underexplored would probably be the mouth, the ear, and actually women's health as well, which are areas that I think, there's been a lot of attention to date paid so far to the gut because we're really starting to understand that. There's been a lot of focus there, but there's other areas sort of the human body that I think will become quite important and that we're building better and better understandings of. And I think delivery to those systems will be quite a key piece as well.
Two additional areas, I think any area not well vascularized is always a challenge.
Yeah.
And then the second is any sort of implant-based infection is a challenge. And then a larger comment is that I think synthetic biology actually holds potential to enhance delivery and requires enhanced delivery to get circuits. So one historical comment is when we first got excited about this space back in 2000 and introduced something like the toggle switch, I remember giving talks back then when gene therapy was popular that we would come in as the control guys once the field worked out the delivery challenges. That field imploded on itself, came back about 5 years ago, kind of unannounced, with still the same delivery challenges. In that both safety and efficacy, can you get enough? Now when they turn to us, they're looking for control, but in many cases, they don't have the right viral vectors that enable us to put the size of circuitry that we'd like. And so I think they're fascinating new challenges, including in the commercial space, at the interface of the two.
We'll take one more question, and then I'm going to just prep the panel. We're moving to the future that Jim was talking about, so I'm going to be asking you about some applications in the long-term future. So think about that while we're listening to this question.
Where are the discovery bottlenecks? Where are you finding challenges, finding new products to take into the clinic and new products, new commercialization products?
Oh, crash of microphones. Yeah, Christine, perfect.
So I think for my area in kind of chemical manufacturing, it's well understood that nature holds enormous wealth of diversity in compounds that have a lot of interesting bioactive properties. But the main issue is in trying to identify what those compounds are, right? And so I think in terms of discovery bottlenecks, it's really how do you create those large natural product libraries that then become available for screening for a number of different applications, whether it be for pharma or for consumer products or whatnot. And so I think, basically how do you access that diversity and how do you find the right applications?
Yeah, if there's directly to that, go ahead.
I would say, I think, we've made a lot of progress on coli as a chassis, but if we want to look to other organisms, particularly that are related to human health, there are a lot of other good microbes out there, but we really don't have the genetic tools to effectively manipulate those. And so building additional parts libraries, additional understanding on in just the sort of the depth of the genomes that we have coverage on, as well as also the vector piece that Jim brought up, which is what's the amount of new DNA that we can actually deliver into organisms is, in my view, some of the pieces if we want this to expand even further sort of beyond the kind of the coli yeast paradigm that seems to be kind of the dominant piece right now.
So quickly, the 3 development bottlenecks I'll put is one is that we still don't have a great ability to put together parts in a defined way that gets you the function in a quantitative manner that you want out. Second is we don't have as many parts as people think and as we'd like, and so we have to harvest. And then 3, which has now become a dominant theme in the field, is that we thought by taking so-called orthogonal parts, so parts that normally wouldn't exist in the bug and putting in a circuit, would allow you not to have interactions in a meaningful way and screw up your circuit. And that's just not the case in the sense that you actually introduce a circuit, you put a significant metabolic energetic demand on the bug, and it's the biggest challenge we have right now is how do you get the bug to stay happy and to have that circuit or pathway do what you want it to do while the bug remains happy.
So please play with me now, panel, because this is typically a little challenging. So if we're looking at the meaningful future, and I don't know what that is, so I'm going to ask this to you in stages, then I want kind of like rapid fire responses, and it's fine if you don't have one, you can just move on, and I'm not trying to push your buttons, but I'm trying to just push a point here. In the next 2 years, name one meaningful innovation, concern, worry, anything that we should be concerned about or excited about, Malik.
Exciting is the microbiome field. I feel there's a lot going on in that space, and since it's a new area, a lot of groundwork needed to be done, but I believe now is the time when we'll start seeing the fruits coming out soon.
Yeah.
Using engineered exosomes to deliver synthetic circuits to human cells.
I mean, I think the focus still needs to be on consumer perception and adoption of these new products that are coming out.
Next couple years, I think implanted cell bioreactors.
I'd say clinical readouts of cell-based therapy, be they actually engineered T cells or engineered coli.
Let's move to 5 years, Malik.
Oh.
Let's call it 5 to 7. I mean, call it 5 to 7. I mean, the next stage.
I think the diagnostics is another field which probably will benefit a lot from synthetic biology developments, and that thereby pharma will also be benefited from that.
Jim?
Engineered exosomes that can be injected into your blood as synthetic sentinels that can sense what's going on.
The academic course has an
I'll
answer to
keep
this.
on
So this
theme,
is not
right?
an NSF. So
Yeah, well,
exactly,
yeah.
I get it. So we'll move to.
You may not hear exosomes again from, but you might.
We're going to move outside your comfort zone soon. Christine.
I don't have an answer. Complex chemistries access to nature.
Vincent?
I would say magnetically charged particles that could go anywhere in your body based on magnetic force. Nanoparticles now.
Okay.
Can CRISPR-Cas really translate successfully into the clinical paradigm?
Okay. All right, so let's make it easy. Let's move to 15, 20 years.
No.
Malik?
No idea.
No idea? Jim?
Evolvable antibacterials. Ones that can,
repeat that?
evolvable antibacterials, ones that evolve in the face of emerging resistance and remain effective in the face of resistance.
Okay.
I think for me, it's the existence of multiple microbial chassis that can produce building blocks used for commodity chemicals, very basically high volume, low value chemicals.
Engineered humans.
Can we figure out what to do with phage?
With?
Phage.
Okay. So let's move to Jim's original time frame, the next 100 years. Malik?
None of us will be here, so why can't
Our kids will be here.
Well, yeah, maybe
Depending
that's
on
something
your-
we can do.
depending on
Immortal
some-
human beings.
Okay. Immortality. Jim?
Biology will still not be an engineering discipline,
Yeah.
but we we will have significantly enhanced our capability to design.
Okay.
So I agree with that, kind of better understanding of the intricacies and complexity of biology and being able to more easily engineer that. I think a really interesting new application that probably will take pretty long to establish is the DNA-based data archiving. I think that's a really hot new topic, and curious to see where that goes.
Vincent?
Yeah. I think, universal genetic profiling, where the entire population is analyzed for genomes.
And I would say I'd hope that we'd be able to make some real progress on solving what I expect to be food, climate, and water crises in the next 100 years, because I think that this discipline actually has opportunity to do that, unlike other areas.
Okay. Is there any burning questions? There is a burning question. Can you turn up the mic?
About 1,000 years. I was just wondering about other disciplines like artificial intelligence, machine learning, digital health. How do you see certain other disciplines coming into synthetic biology, and how do you see those two collaborate and strengthening each other?
Who wants to take that up? Jim?
So we've engaged in a number of discussions this the last 6 months, and where I think it can help is, can one use machine learning and even deep learning? So deep learning, I think we can actually generate the data sets that could be meaningful to enable us to infer design rules. And so can you go from sequence to function? So you can envision doing that quite simply with the RBS calculator that Chris Voigt's group did. So where on base sequence you can actually get to function. Can you go further? You can do it with the ribose switches. Can you now get to protein from sequence on the same function? Can you go to circuitry? Can you go to pathways, synthetic circuits? I think there's great potential there to really now expand our ability to design directly from all the AGTCs sorts that we heard from Asimov to getting to functional components. And Google's very interested in this space.
And I would agree there, but I think it's also a question of, can we get to the scale of data that will allow us to actually build the algorithms that are going to give us the right output, right?
I think if
That's a
Google's
good point.
interested in it,
Yeah.
it can be done.
Yeah. But we
So
still
unfortunately,
need-
Google can't
right,
generate the
but
data.
It can't generate the data,
And
right?
so they are reaching out. But
Exactly.
George Church can generate the
Right,
data.
right. As
So we have George to
Right,
charge.
or some of the companies, right? So
Yeah.
I think, Ginkgo advertised last year that they made a huge buy from Twist, which was, I can't remember the number of zeros on the
Yeah.
base pairs, maybe, but, they're going to build and test. There you go. You're going to build and test all those parts. You're going to track those. If you've got a database and a software that can scale with that, maybe you can go in and actually build some of the learnings out of that. That'll be proprietary to them. So we have a question of whether we have public access to it, right? But,
I think related
someone
to
will do
that,
it.
I think actually synthesis probably is not going to be the answer because it's too expensive.
yeah.
But I think generating diversity
Yeah.
through large-scale mutagenesis and then be able to select for a function
Oh,
and
that's
couple that
interesting.
sequence
Yeah.
is
We
going to
sort
be
of design those
economically,
libraries
so
first, yeah.
you take advantage of biology, which we have not done as a field, which is biology evolves and biology likes to reproduce. We take advantage of the reproduction, but not the evolved aspects, is where I think we can get after the data size
Yeah.
that deep learning will need.
I think the other really important way in which computational can assist us here is not just on the design component of biology, but basically understanding how biology works. So it's really leveraging these large data sets that are generated through proteomics and metabolomics in order to really understand what the underlying biochemistry is. Because having that understanding of the genotype-phenotype relationship is what will allow us to advance further.
Malik, any thoughts on machine learning?
I feel going on those lines that we were mentioning, with all this genome sequencing, human genome sequencing companies that are there, if the new generation of kids, like whoever is born, gets a genome map, then that will initiate a lot of machine learning. We'll have everyone getting sequenced, and based on that, we'll be able to predict a lot of diseases and patterns. So that's where I see.
So lastly, and we really have 40 seconds left, but if you have a challenge to some existing MIT engineers, or really anybody who is developing companies out there, what should they be focusing on right now?
Building the best team you can. And it's all about team. Unfortunately, it's not about my technologies, it's about the team. So all the successful companies that I'm involved with, it's the business folks, the science folks that team up. It's not technology. So come to networking events like this.
It does help.
It helps, but it's 5-10% of the story. It's getting your team together.
And I think on that note, I want to just hope that we can thank the panel for this extraordinary contribution. Thank you very much. And as we're wrapping up, please just sit for 1 minute, be patient with me. I just wanted to announce two different things. So first of all, thank you to all the rest of the speakers. This has been an extraordinary turnout, I think, and we were very pleased to see what's going on in the field of synthetic biology and new materials here at MIT and obviously beyond with our partners and corporate activity. There's two events, talking about events, that you should know about if you don't already. Next week we have a health and sensing conference that brings together lots of interesting technologies in health, and that's going to be on campus, so exciting to think about attending that one. And as I previously announced, we have a Cracking Cancer 2.0 event in December, similar format to this, and as you note, it's the second one in its in its turn, and I think the idea here is, some people told me, well, why do you do another event on cancer? Well, I think the situation is a little similar as with synthetic biology. What was news last year is not news this year. I mean, there are just so many, even of some of the same startups that are telling very, very different stories this year because the field is just moving so rapidly. So that will be another interesting place, I think, to discuss synthetic and non-synthetic biology and other topics, that are happening in and around MIT as regards cancer, technology, innovation, startups, all of that. And with that, I wanted to thank everybody for coming and also invite you to, stay here for a little bit and network and partner with us. And if you are left here without knowing how to get hold of us, we haven't done our mission right. So I want to again repeat that on the corporate side, there is Cheryl, there is John, and there should be Eric, but maybe he's out there talking. And you can also grab any of us, Marcus or myself, Tron, if you're interested in the startup perspective on things. And with that, thank you very much. Have a wonderful day. And it seems like synthetic biology will not remain a secret for very long. Thanks.